Welcome to this episode of Life Science Success. For those of you who don't know me, my name is Don, and I'm a digital marketer in life sciences. And before we get started with our interview today with Markus Gershater, I just want to provide just a little bit of information about my digital media marketing business, who is our sponsor today.
This episode of Life Science Success is brought to you by D3 Digital Media Marketing. Dive into the digital world with confidence and creativity on your side. D3 Digital Media Marketing is your all-in-one solution for making a lasting impact online. From the vibrant, engaging world of social media marketing, podcasts, and webinars, to the strategic depths of website development, SEO, pay-per-click, email, and content marketing, we tailor our approach to fit your unique brand story. Let's elevate your presence, connect with your audience, and drive success together. Your digital journey starts here. Visit d3digitalmedia.com to learn more.
So with that, let me just introduce our guest, Markus Gershater. He's the co-founder of Synthace. Markus is a visionary in the field of synthetic biology, dedicated to transforming life science R&D through innovative AI and cloud-based platforms. So with that, welcome, Markus.
Thanks, Don. Yeah, nice to be here.
Yeah, it's great to have you here. And one of the interesting things with regards to synthetic biology that I always find is, one, I feel like there's a definite evolution that is happening, and I could only imagine the world that you live in. And secondly, there seems to be a lot of overlap. So I do look forward to, just getting into, how is Synthace, different, and those sort of things as we go through. But before we get to that, could you just take a minute and introduce yourself a little bit more to the audience, as well as just talk a little bit about what brought you to the field of life sciences?
Okay. Yeah, well, I've always been utterly fascinated by biology, like for as long as I can remember, to the extent where I would often be quite baffled as to why people would be interested in anything else, with that kind of blinkered view of the world that I guess many of us might have had when we're young. So I now have more of an appreciation for other disciplines maybe, but I still have that abiding passion for biology as one of the most complex and fascinating phenomena that you could choose to study. So that's why I'm in life sciences. Originally, is just because I could never imagine really doing anything else. It's not a particularly interesting answer, but that's where it is. And then really, I haven't had any specific part of the life sciences that I wanted to be a part of or be involved in. But my kind of career is just, been a sort of mixture of enthusiasm and opportunism in different areas and seeing what opportunities come up and making the most of them, really.
Yeah, I feel like a lot of us, I mean, there's, you could say that there's a defined path that I set out, that I absolutely, wanted to know that I was going to do this, then this, then this. But the reality is many of us are sort of, at the mercy of life as well.
Yeah,
Back to
that's
biology.
that's where a lot of it can be fun, right? Is if you have a really clear path in life, then that's assuming that you know all the different paths at the outset, potentially, which obviously none of us do. So I think sometimes, I did have that abiding passion throughout the whole of things, but that's pretty much the only constant. I started out with a PhD in biochemistry, and then worked briefly in industry at a company called Novactor Biosystems, where I was essentially looking to engineer biological processes, right? So all of a sudden you go from complexity being the thing that fascinates you and intrigues you to complexity being the thing that's getting in your way, right? As the thing which is preventing you from being able to actually make something really robust and effective out of the biological system that you're working with. And then from there, I went back into academia because I saw a great postdoc that came up at University College London. And it was while I was there that we co-founded founded Synthace. So I've sometimes been asked to do kind of careers talks and all this kind of thing, and I don't know how helpful you can be really, except for just saying, hey, just look at the opportunities in front of you and go in the path that seems to take you, the most. That's been my And then I've been really fortunate, I think. I mean, it always has to be acknowledged. If you end up in a situation where you get to do what you love on impactful things, then I think there has to be a bit of good fortune in there as well, right?
Yeah, I feel like, yeah, I mean, especially whenever it comes to fields like biology or life sciences, I feel like so many of us, sort of run into the same people over and over again. There's a lot of the same sort of space that we're all playing in, but at the same time, the field is so broad. I mean, there's so many things that you can do in life sciences. So you could decide to go down one track, and that leads you down, a totally different set of things that you could possibly do. And
Right.
obviously, to your point, it needs to be interesting at some level.
Yeah.
So, yeah.
I mean, I'd like to think that there isn't much of life sciences where you're not going to find something interesting. But like again, just to its complexity, obviously that means that there's so many areas that you could go in very deep specialism in any one of those, right? Um, and I'm not sure that really suits my personality, though. To be honest, I'm more of like a systems thinker, I guess, as opposed to like someone who really gets down into the nitty gritty details of things. I really value being able to work with lots of different kind of ways of thinking,
Mm,
right? Different people with different ways of thinking, because it's in that kind of synthesis that you really start to get to grips with the problem.
yeah, absolutely. So can you tell us a little bit about what motivated you to co-found Synthace, and what how has your vision evolved since the company's inception?
Huh. Um, so I guess I alluded to it earlier. I did my PhD in biochemistry up in Durham University, and like a lot of PhDs, it was a bit of a slog, right? And you're trying to sort of chip away until you get enough results together, and you kind of hope that you're going to find something of some kind of um, conclusiveness, so that you've got something that you can write up and publish. Um, and then I went to this company called Novax Biosystems, and essentially what they were doing there is the bit that I was working in is doing biotransformations. So this is where you're basically doing chemistry, but with biological systems. So that's either an enzyme to catalyze the reaction, or even whole cell systems where you put in a substrate, and then it gets converted by that microbe into whatever the outcome is that's needed. Um, and so then, not only do you have all that complexity to deal with, but you don't have the 3 to 4 years of time to chip away to make it work. Um, and I was challenged quite early on with a process which had been developed in an academic environment, which I then needed to industrialize. Essentially, this is what the Novacta role was within that particular consortium. And I had 4 months to do it in, and I needed a robust and high yielding process out the other end. And I sat down and worked out all the different things that I might want to investigate in order to do a rigorous job on this, right? As an ambitious young scientist, I wanted to make sure I was doing the best possible kind of industrial job that I could, because I was just new into industry. Um, and you start listing all the things that could affect your process, and you keep listing and you keep listing, and you realize that there's way more things there than you could ever hope to investigate. Um, and so um, I basically I was very fortunate, because actually my dad originally worked in the field of process development, and so he's got a lot of experience in it. And he'd been talking to me about multi-dimensional ways of experimenting, so testing many things simultaneously. For a long time, it's a process that's often referred to as design of experiments, and he'd been trying to convince me that I should be working this way for quite a while. And I was kind of like, yeah, he's my dad. What does he know? And so I'd plugged away at this PhD largely without thinking about that at all. But then when I was in this industrial setting and I had this deadline and a complex system to work with, I was like, maybe the old man might have known something after all. So I picked up the phone and basically said, "Oh, Daddy, I've got all these factors I need to test. Can you teach me DOE?" And he said, well, not over the phone, I can't. But like, long story short, basically, I started using this methodology where you're looking at many different hypotheses simultaneously. And it turns out that it's super powerful, right? And it's not a particularly new methodology. It's been around for quite a while, but it's one which is largely neglected within the biosciences, unfortunately, because the biosciences is all about that multidimensional space. It's all about like how like many different things can interact together to then generate the emergent phenomenon that is life, right? That is what biology is. And yet we look at it like in a reductionist way of thinking about, okay, what's each of the parameters that we want to check out. So when you start looking at it in this more kind of holistic way of like looking at lots of different parameters simultaneously and the way that those parameters interact together to give the biological system, all of a sudden it's like you're using a tool which is actually suited to the phenomenon that you're studying, right? And so instead of like laboriously having to inch by inch make progress and 2 steps forward, 1 step back, ambiguous results, things that work one week but don't work the next, all things that are really familiar with anybody who's worked in a bioscience lab, right? Instead of that, all of a sudden I had this feeling of, oh wow, I actually understand what's going on in my system. I know where I need to move next. I know how I need to iterate. I know what conclusions to make. I've got all the statistical significance backing stuff up built into this experimental design in the first place. And long story short, for that 4-month process, I managed to improve the yield of that process 7-fold in 3 weeks.
Wow.
And they were tough weeks, like there's hard work. And
Yeah.
I'd love to say this is my genius, right? But it wasn't. It's just because it's actually using a methodology that is much better suited to biology than the sorts of experiments that we are trained to run, right?
Right.
So like in that way, it's quite simple. Anyway, so that basically means, hey, I could now see that maybe the ways that we are doing biology aren't necessarily the most effective ways, right? This is a really, a massive, like startling kind of jaw drop moment that I had of like, oh, wow, the way that I've been taught to do biology doesn't necessarily get us where we need to go. And that's a bit of a moment which is both exciting and sort of frustrating simultaneously, right? Because it's like, well, hang on a second, I mean, DOE's been around since the 1930s, right? It's not a new method at all. And it was actually developed first in the biosciences. It was developed on an agricultural research station that's just north of here. So I'm in London, and there's an agricultural research station called Rothamsted. And in the 1930s, there was a statistician there that worked out, oh, we could do much more effective experiments, right? So, when I did my postdoc at UCL, it was with the knowledge that actually we can do things fundamentally differently and we can do things fundamentally more powerfully. And so then when an opportunity came up to start a company, it was with this at its foundational heart, right? So yes, we called ourselves a synthetic biology company at the time, and we certainly were in a lot of ways. The definition of synthetic biology is an interesting one. I tend to think of it as basically the intent to systematically engineer biological systems, right? And actually in the UK these days, it's often referred to as engineering biology these days, which I think is maybe a better name for it. But here was this methodology of multi-dimensional experimentation, which did give predictivity, which did help you to understand the fundamentals of the system you're working with. And then at the same time, when we first founded Synthace, it's when like the ordering of new genetic parts, the new of new bits of DNA was becoming routine, right? So
Right.
all of a sudden, we could just get whatever DNA we needed to try and engineer a system. And so you combine these two things and you basically have, okay, so we can look at all the different ways that maybe we can grow cells, all the different things we can feed them and that kind of thing. But then at the same time, we can systematically vary the genetic components within that organism, right? And so then you can see how the genetics interplay with the different environmental factors that you're imposing on that system to try and then get the phenotype that you want, right? So, genetics plus environment equals phenotype. So it's a way of trying to systematically explore biology such that you can get a particular biological outcome. And that was kind of that. It was that the promise of that kind of way of working, and the evidence I'd already had around a lot of it, which kind of motivated us to say, okay, yeah, there's a new venture that might be able to make use of this, and there's a good reason to start something and see what we can do with it.
I'm glad to hear too that, I mean, I just to kind of rewind a little bit of the story that you told. I my father, I know a lot of times whenever I would talk about problems that we were seeing at work, he constantly, I mean, he had a quality background and had a deep statistical background as well. And he constantly would sort of inject himself into the problem and sort of say, well, this is how I might consider, solving that particular issue. Some of the, I mean, in my past, I've run into some people that have really sort of taken on DOE and really made this a part of their life,
Right.
and I feel like the more that you can sort of understand the parameters around leveraging DOE, the better you can you know use it to really design something that in the end will do what you want it to do. And to me it's absolutely amazing. But at the same time, going through my Six Sigma training, we had to use in the classroom, we used a toy catapult and built
Right.
a foil ball and had to predict like what would the actual landing point be for the foil ball. And not certainly as complex as what you were trying to do, but at the end of the experiment, we were able to say, okay, so here's exactly where this thing's going to land, the trajectory, and roughly how we need to set the catapult to be able to hit the spot that we're aiming for. So an amazing tool set by far.
Yeah, and it's one where the importance of kind of getting over that hurdle of, oh yes, this is something that's really useful, like or the importance of some kind of practical demonstration in that is paramount, I think. Like, if you just hear about DOE in the abstract, then it's just like, sounds like a lot of dry statistics and that kind of thing. And I mean, I've got a reasonably mathematical mind, but I never really found, just the theory of statistics particularly interesting. But then it's actually one of the most exhilarating things you can do when it's actually applied, right? And when you actually make progress with what you want to do, whether it's, getting your catapult to be a lot more accurate, or engineering a biological system to produce a high value or a high complexity product, right? And I think the other difference between the systems is that with the catapult you could imagine, you could do one factor at a time and not expect there to be any particularly deep interactions between those factors, and you could probably get to a reasonable answer. Whereas with biology, if you do one factor at a time, the only way you'll get to a correct answer is through luck.
Mm.
Because the amount of interactions that are there, it's an emergent system, right?
Right.
It is interactions. That's what biology is. And so if you're not doing experiments that can tease those out, then you're really just trusting to luck. And then if you look at the progress that we make within biology, it kind of fits. It's like, yeah, actually, it is really tough. A lot of things do fail and work one day and don't work the next. And and it has all the hallmarks. That like experiential sense of being a biologist has all the hallmarks of using underpowered tools. Essentially, it's like and we always put it down to, oh, because biology is complex. Of course my experiments failed. Biology is complicated. And of course we've got ambiguous results. It's a really noisy system, or whatever, right?
Right.
um, and this is what we fall back on, because otherwise it would be completely soul destroying. It's like, if we took it all as a personal failure every time a biological experiment didn't work, then like none of us would stay biologists, I don't think. And yeah, so it is a real exhilarating feeling when you actually have a system that can then actually make decent progress in it. And early days of Synthace, when we were this biology company, we were using things. I mean, in Novactor, I got a 7-fold increase in 3 weeks. One of the case studies that I still use today when I'm talking, I get to lecture a bit about DOE and stuff, and have some fun and enthusiastic rants in front of some undergraduates about why they need to be thinking about how they're going to do biology when they become scientists. And one of my favorite case studies is still one that we did early days of Synthace, where we looked at 15 factors simultaneously and got a 200-fold improvement on literature precedent in 5 weeks. 200-fold, right? That's what, 20,000%.
Yeah.
And those kind of things, they make you pretty evangelical. It's like,
Right.
like there's no going back from that kind of thing. But fundamentally, like, there's one thing about having a really powerful methodology and technology and know-how, and then there's quite another about then actually making that into a really successful commercial enterprise, right?
Mm-hmm.
And so it's all well and good. Yes, we could take biological systems and engineer them really well, although there were some that we failed on as well. Like, if you don't have a good place to start with an optimization, then there's nothing to optimize, right? So
Right.
you have a start point and a good assay and a good experimental design, and then you can make progress. But so we were, but when it worked amazingly. But then monetizing that is then a real challenge. Like, we were essentially looking to make products and sell them. This was our first idea. Very simple. Like, use the technology, make efficient processes, make stuff, sell the stuff. But then actually, it's a lot more about market access and route to market and that kind of thing than it is about the actual technology, right?
Right.
And so it's all those learnings of kind of young and maybe naive entrepreneurs, of like, oh, actually, there's a lot more to this than just making the technology work in the first place.
Yeah, it's definitely is a challenge shifting from research to a product as well. And I think maybe this is a good transition point as well. So one of my questions is, how does Synthace help to transform life science R&D, and what are some of the key innovations that your platform offers? But also, maybe could you just provide a little bit of insight in that, what led you to that point, in terms of bringing it to market as well?
Right. So the common thread, I guess, is biological complexity, and then what are good ways of addressing biological complexity, right? So we've already spoken about one of them already at great enthusiastic length. People who know me know that this is very common, that I'll be talking, I continually get teased within the company about, like, how often I can just turn the conversation to DOE, right? But then there are other technologies and methodologies as well, which we turned to in those early days of Synthace, which sort of are multiplicative when you combine them, right? So there's DOE, and you can do it by hand, right? And those early examples, the one that I did at Novactor and the early ones at Synthace, they were all done by hand, and they're incredibly tough to do.
Right.
Like, really, really grueling. I mean, in Novactor, I did these 32 run experiments, which is already quite a few, and it was all in shake flasks, and I was kind of like arranging them on my bench and trying to work out how to keep them all straight in my head, and with the experimental design. At Synthace, then we were doing 64 run experiments and upwards. And this is where each one of those experimental runs can vary over like 15 different factors from each other, right? Like, they're just hugely complex. And we worked out various kind of lab-based coping mechanisms for trying to make them work. But fundamentally, we realized that really the most powerful way of doing these kind of experiments, and frankly any experiment, is to automate them, right? So, and this is not something which is an insight that's unique to Synthace by any stretch. I think everybody kind of appreciates how automation can have a lot of potential in the life sciences, and has already delivered a lot in the life sciences in certain domains. But then the problem is like, yes, automation's great, but DOE and similarly powerful experiments are hugely complex, and so programming into that device about how to actually do that experiment is really tough. Like, If you do it just at the level of, moving liquids about, then it will take you, maybe a week or two to actually program the device. And then you run the experiment once and you never run the same experiment again. And so then, like there's no return on investment for all that programming effort. And so we immediately looked at, okay, how can you automate the programming of the automation, right? So how can you take an experimental design and automatically work out the plan for how that design needs to be carried out, and then turn that plan into automation instructions so that the automation will actually do it, right? And so this was all internal in-house capabilities that we were doing to kind of push the envelope on the kind of experiments we could run. And then for various reasons, and part of it was about like, trying to find a business model that really worked. But also that we realized that the moment you're talking about software, then the impact on the world that you can have is dramatically higher, right? If instead of just using it as internal software to, make Synthace work, what if you could then make it into a fully productized thing, which then you could put out to the life science industry overall, and help like anybody who needs these methods to use them. And long story short, that's the very deep pivot we did, going from being a biology company to being a software company. And the platform itself, so the Synthace platform, I mean, we've spoken a lot about DOE, but really what the Synthace platform is a way of planning and running experiments that is like fully digitized, right? So, and what do you need to be able to do that? Well, you need a way of telling the system what experiment you want to run. So there's some kind of definition, and we basically have a drag and drop interface where you connect together different building blocks into like a workflow, essentially. So, you do this step first, and then the output from this step wires into the next step, and so on. And this is very much at like the sample level. It's right. We have these samples. We want to take these ones and dilute them like this. We want to make replicates like that. We want to mix them with this reagent and so on. And so it's building up that kind of logic, but you're doing it in the digital world from the outset. And so then Synthace has a planner at its heart, which then works out, okay, you've told me your intent. And now this is the specifics of exactly how, all the liquid handling actions that have to happen in order for your intent to become reality. So it's taken from that high level definition to a very low level, very granular plan of like, 3 microliters of this liquid has to move from here to here and then be mixed in this specific way, right? And then it converts those instructions into automation instructions, which can then be run in a lab. And then because we know exactly what's happened in that experiment, because Synthace is the platform that's actually defined all the actions, right? So we know all the actions. That means that any data that's produced can be put immediately into the contextual metadata, i.e., like, all of the experimental plan which went into producing those data. So it means that, when you've worked with Synthace, you have, the full process recorded in a huge amount of detail as to exactly what went on, and all the data is put into that context. So you can like auto structure the data essentially that comes out the other end. And then one of the most powerful outputs of this is being able to do multidimensional experimentation, right?
Right.
Which is where we do DOE.
Very nice. I mean, I know how difficult it is though, transitioning a company from a very research intent, company, and then productizing that and finding the fit. In terms of the, kind of the journey that you made in that, I would imagine that it wasn't all just inward facing, you sort of looking at how processes or biological systems are developed, but more or less, also involving people that would be in the end, customers as well. How did you sort of make that transition?
Yeah. I mean, I'll be honest, not nearly as resolutely as we should have done in retrospect, right? We had really clear vision from the outset that, that there were better ways of doing biology, and that, like this combination of digital tools plus automation plus multifactorial experimentation, can be truly transformative. And actually, to our credit, there's nothing that's happened since that which has made me think anything otherwise, right? It's absolutely amazing seeing the kind of experiments which our customers now do with Synthace. They take it, and they take it still a step well beyond what I could have envisaged, which is fantastic. But when you have such a strong internal vision, that doesn't mean that you shouldn't combine that with a lot of just very detailed research into the realities of the life which your potential customers are actually living, right? Um, and and that's particularly acute when you're asking those customers to make quite a radical change to the way that they're doing their science, right?
Right.
And so, again, I think I hope that most people's entrepreneurial journeys are ones where they make mistakes, and along every step, because that certainly feels like the journey that I've been on. But it's one of those things that I definitely look to change, and is about the degree to which we looked to tailor what we had to the realities of the way people work in the lab. Um, it's it's been a hard one learning, and these days, we have our scientists go and sit next to scientists, you know because my team in Synthace is still the lab team, right? We still have a lab, because we need one to be able to make sure that the software actually makes robots run in the way they should, and actually produces good results. But the other way, things that our biologists within the company do is they go and they really learn about the science that people are doing, and make sure that actually the platform is set up in such a way that it can deliver on exactly what those scientists need, as well as potentially kind of nudging them a little bit to kind of say, oh, and this experiment, actually we could do that in a much more powerful way, right? So you're kind of sort of continually trying to um, move the thinking on a bit
Mm-hmm.
about like the way that we approach our science. um, so it's not a small undertaking that we've embarked on, but it's one that feels very meaningful. But yeah, going back to your original question, um, I think um, yeah, if you reran the tape, I'd be looking for us to spend an awful lot more time with scientists and their leaders, in those early days when we were first, transferring over to being a software company. And you could talk about all of the transitions we had to make in terms of hiring software engineers and all this kind of stuff. But actually, the biggest transition that we've done over the years is really understanding about how to make a product, and not a physical product. If the original business model had succeeded, then we make a physical product, we make sure it's high enough quality, and we can make it in the quantity and yield we need to, and then you ship it, right? It's like market access is an issue, but unless you're bringing something radically new to the market acceptance and that kind of thing isn't such an issue, right?
Right.
But with a software platform like this, it's all about um, making it so that it fits within people's lived reality in the lab.
Yeah. So my people that listen to the podcast know this, but the my background is much more in like business operations and digital marketing. But so when you look at the way that people are doing experiments today, how often do you feel like AI and machine learning is being integrated into the things that they're doing, naturally, and then how much of it is, people are still using, older methods to continue to develop things? I mean, is AI and machine learning much more readily available, I guess, for everybody, and are people using it? Is the question.
I mean, it's a landscape that's changing all the time. I would say for the people that we see the most, i.e., lab scientists, I'd say that AI is almost non-existent, right, in what they're doing. And but then you look at the way that pharma companies are transforming how they're doing discovery, for example. Then you see a lot of AI there, and actually being used, with some really exciting success. So for specific use cases within the value chain, I think AI is already starting to show, this latest wave of AI, I guess, is really starting to show how valuable it can be. But I think there's quite a long way to go before it's a kind of, well, a sort of ubiquitous tool which people have in their armory, right? And I mean, when we talk about AI, it's a bit of a nebulous concept, and because actually it encompasses so many different tools and strategies. Um, and I think over time, what we'll see is just more tools suited to specific purposes being deployed in different parts of the value chain and different parts of the discovery process. And so, as each one of those starts delivering, then we'll see an overall transformation of the space. And in the same way as I was referring to DOE being a tool that's just dramatically better at working out biology than like more sort of traditional ways of doing experimentation, I think that AI holds the promise of, like, of all disciplines. I think that biology is the one that needs AI the most, right? And that AI will be able to help the most. And this isn't just hype. It isn't just it's because, again, look at the nature of biology and look at what AI is good at. AI is very good at inferring underlying patterns in very, very complex systems with huge amounts of data, right? And in biology, very, very complex system, we can generate huge amounts of data, and there's no way that human intelligence will be sufficient. No way that DOE will be sufficient by itself to unpick all of that. And so the prize is massive, and it's worth going for. But to answer your original question, yeah, it's got a fair way to go, I'd say.
Yeah, I mean, even just in sort of the AI models that are out in the public, right? I mean, there's a variety of people, I feel like, that use AI for different things, but then there's certainly other people that just haven't found a use case, or think that it's some sort of technological hype, and so
Yeah,
maybe they're not taking a look at it, but it will catch up with all of us, because I feel like it's getting built into everything that we have. So yeah.
yeah, and it already was, maybe even well before, people really thought about it in that way. When you ask Google Maps to tell you the way to get somewhere, like, okay, you might not realize that that's got some algorithms in the background, and some models in the background that are telling you the best route. But that's nobody uses paper maps anymore, right? So I think yeah. I mean, it's like any other kind of tool. I think what can be really helpful is, there's a um, I don't know exactly what her role is, but she's kind of like a, a sort of policy stroke ethicist, who I've come across before in the synbio space, who's also written a lot about AI, called Hilary Sutcliffe here in the UK. And she has something really helpful, I think. And she says, whenever you hear the term AI or read the term AI, just mentally in your head exchange it for software.
Mm-hmm.
So not, AI is able now to understand natural language and, convert it into, instructions, but software is now able to understand natural language and convert, I think it strips out a lot of the mysticism about it. And I think that's kind of what we need, right?
Right.
Because it is this, it gives you a much better sense of the pragmatism around it. And then, you want to see how powerful it is. Well, give it a go. You know, I mean,
Right.
anybody who has an opinion about ChatGPT who hasn't actually used it, right? I mean, it's the way that you can really learn about where its strengths and weaknesses are and understand it as a tool, right? It's funny. We've just had some job applications recently, and I was just chatting with uh, one of my team, and he was saying that we had some applications where not only had they generated the cover letter using ChatGPT, but they then hadn't taken away the comments that ChatGPT has at the bottom. Basically, just saying, if you use a cover letter like this, it will emphasize all your strengths and, like, put them against, like, the. So it's like, and the funny thing is that if they'd used ChatGPT well, and
Right.
they're generating a cover letter such that we couldn't even see it. Well, great. It shows that they can use, but the fact that they used the tool but used it badly, that kind of reveals a lot. So yeah, there's huge potential for it. But I think, for it to really take hold, there's a lot of things that are going to have to happen. One thing, the tools will have to be both relevant and accessible, obviously. But the main thing is about the data for me,
Mm-hmm.
right? I went to a conference recently, and I was in an AI track, and um, I honestly, sometimes I feel a little bit fraudulent in those kind of tracks because I'm much more about like, experimental design and this kind of thing rather than, like if you ask me which AI algorithm to use on something, I won't have a clue, right? So I know very little about the actual mechanics of how machine learning works. But I was in this track nonetheless, and there was a guy who was talking before me, and he just had this fantastic slide, and it was like, to the kings and queens of AI, data is God, right? And I just love it, and I've been stealing it ever since. It just, what AI is really good at is taking these huge, very complex data sets and trying to make sense of them.
Right.
But it needs the data, and it needs the data to be both comprehensive to the thing that we're looking to learn about and of very high quality.
Yeah.
It can interpolate, but it can't extrapolate so well,
Right.
right? So if the thing that you're trying to learn about is outside the realms of what the AI has been trained on, good luck.
Yeah.
It's not magic; it's software, right? Um, so um, like, it's like when you know when people expect LLMs to somehow be magically intelligent about things that aren't language,
Right.
right? Or that can't be inferred from language. It's like, when you try and get an LLM to play chess, it takes its own pieces, right? Or or
Yeah.
if you look in the lab, right, then it doesn't understand how a lab works. It's
Mm-hmm.
going to do like the equivalent of trying to, pour a pint into a shot glass, right? Because it doesn't know the physical constraints of what you have to do there. So we've been looking to integrate, like the power of things like LLMs into the lab. But what you need to do then is you have to have a system like Synthace, which has all the models of how the lab actually works, of how liquids can be moved and how they can be mixed. And that can then hold the LLM to account, right? So when the LLM comes up with something that is just nonsensical, you can have that feedback loop and say, no, go try again. Um, and the exciting thing is that then, what you can then do to define a new experiment is you can just say, hey, I want to run an experiment. And it's like, oh, what kind of experiment would you like to run? Oh, I want to do an assay optimization. Okay, when doing an assay optimization, you need to think about these things. Ding, 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 ding. Can you give me, like and so instead of it being a process where you have to think about every detail off your head and do a lot of planning yourself, and then like digitize that, like that concept in your head, you can actually have this kind of interactive way of defining a new experiment,
Right.
which is amazing, right?
Yeah.
Really, really exciting. But yeah, I mean, there's still a lot of work to be done there, but I think there's a huge potential for this kind of thing to transform how lab work's done. And then when you make those amazing multidimensional data from running very sophisticated experiments, then what an amazing data set to then train future AI on. If you've developed 50 assays in your assay development department this year, then what can all those data sets tell you about the 51st?
Right.
Right. But like, do you want to do that as a human? Hell no. I want AI. I want software to help
Right.
me with that.
Exactly. Yeah, absolutely. So let's talk a little bit about the market potential for this as well. So how does Synthace improve the ROI on pharmaceutical innovation for labs that are operating as a business?
So the magnitude of the change is that if you go from running, I guess, at one extreme, manual, one factor at a time experiments, um, where you're basically thinking, as my experience as a scientist, what factor should I test next? How many replicates should I do to try and address biological noise, et cetera, et cetera. And I'm going to do that experiment this week, and then I'm going to get those results, and I'm going to do another experiment next week, and so on. When you move from that kind of incremental experimentation, one factor at a time, to multifactorial experimentation. That's already a step change. When you then incorporate automation on top of that, that's a multiplicative step change on top of it. I only preamble this because the claims I'm about to make are a bit like they sound like just hype when actually they're just science, right? So we've seen that when you go into functions like assay development in early discovery, so all discovery programs rely on assays, right? Until you have an assay, you don't have a program. And so assay development is absolutely critical. And typically it takes about 9 months, give or and it varies a lot, but on average it takes about 9 months. And this is on the critical path for drug discovery. And also the quality of the assay that you then produce will have an impact throughout the whole of the rest of your preclinical drug discovery, because that's the way you're producing data, right? We have shown that if you do automated high dimensional experimentation using Synthace, you can slash the time taken to develop a new assay by up to 70%. So this is months taken off the critical path. And the quality of the assay that comes out can be dramatically better. So it means that so they use a measure called Z prime, which is basically like a very stringent signal to noise measure. And we've had it dramatically improved Z primes, dramatically improved assay windows. And incidentally, because you understand the system so well, you can also see the bits you can strip away. So often you can get rid of like large amounts of expensive reagent, or use the minimal amounts of an expensive reagent. So you can do it at lower cost as well, right? So this is why I had to preamble with why this works, because I'm now claiming you can reduce time, you can get dramatically higher quality and reduce costs, right? And usually you have to pick between those. But it's the difference of using tooling and methods which are dramatically underpowered for the system you're working with to actually using something that's actually appropriate, right? Or even, dare I say, powerful, right? And so in terms of the actual, figures of how much that's worth, it's very difficult to calculate. You could do some basic calculations. So there's a great tome that's published by the NIH about assays in discovery, the Assay Guidance Manual, I think it's called. And I think it runs to about 1,600 pages now. But in the first chapter, it has a drug discovery process itemized out. So you can actually see proportionately, this is how much would be spent on assay dev. This is how much would be spent on running assays. And you can do some, relatively conservative modeling using that for a big pharma, and and you model in the savings that I've just described, and you can be just in savings alone, anything from 5 to 20 million a year per pharma company. And then you think about, okay, but then what's the cost benefit of getting the drug to market quicker? What's the cost benefit of having less ambiguous results as you go through the discovery process? That's the bit that's really hard to model, right? In terms of value, there's the savings, and then there's the value. So where it's possible to get that proper transformation of a function like assay development, like, actually, have those scientists now doing science in a dramatically more powerful way, then the value is massive. And it's just the start, right? I think the ultimate objective is, all well and good if you can improve preclinical drug discovery, and improve the efficiency and the efficacy of that. Brilliant. We all want to do that. But the main thing is, and there's I was reading a great review article by a guy called Jack Scannell a few weeks back, and he had done the modeling of the importance of having of your preclinical models, so either your cell-based or animal models, the importance of the correlation of those models to actual clinical outcomes, right? And obviously that's important. We all know that's important. Like, there's no point doing experiments if they don't have some kind of relation to the clinic. But what's amazing is that actually, if you just improve that correlation, so correlation coefficients are measured between 0 and 1, right? And if you improve that coefficient by just 0.1, that's the equivalent of increasing your throughput of the number of candidates you test by 10 to 100-fold. Right. So like it's like something that seems like a really subtle change.
Right.
But it has an absolutely immense impact on your chances of actually succeeding in the clinic, um, disproportionate to what you might, intuitively think, right? So the real goal, the real prize here, I think, is using this kind of methodology and combining AI in there as well to make the most clinically relevant cellular models that we possibly can. Right. So taking patient-derived cell lines, converting them into stem cells, and then into highly complex 3D cell tissue culture called organoids, which can then give us the best possible model of what a particular drug might actually do to for patients in the clinic. If we can do things of that kind of complexity routinely and efficiently, then we're not just impacting on preclinical research, but we're actually impacting of the overall success of the overall process.
Yeah.
And that for me, I think, is the ultimate prize for doing this kind of transformation of wet lab experimentation. It's about doing way more meaningful experiments, which can then mean that therapies can be discovered way more reliably, and frankly, therapies that might not otherwise be able to be discovered. I mean, that's quite an aspiration to go for, but I think it's actually within reach, which I think is really exciting.
Absolutely. So, last few questions, primarily focusing on you. So, what inspires you?
What inspires me? I think it's the idea that we might be able to understand and work with biology in a way that might have seemed utterly impossible. Right? I think that biology is both beautiful and complex, and also incredibly brutal.
Mm.
Right? I think the majority of us will unfortunately have experienced that brutality. Like, it has absolutely no sense of justice or what is right. Like, people can just be taken from us in the most arbitrarily brutal way by biology. Right? This thing that I've been talking about as this wonderful thing all the time, it is also utterly brutal. And the idea that we can somehow start to get some semblance of control, some semblance of ability to actually understand these systems and work with them such that, like, it becomes dramatically less brutal. Yeah, that inspires me.
Perfect. What concerns you?
Wow, loads. I think we've spoken very uh, enthusiastically about, like, all of the changes that might be possible and the benefits that could come from those. My concern is about actually adoption of these things, and, like, and how quickly we can make that happen. It'll happen, but how quickly will it happen? And you know can we make it happen in such a way that it's as beneficial as possible to as many people, right? When we're talking about things like AI, it's well documented about how pretty horrible that tool can be and all. You know, I the kind of biases that are baked into LLMs because of the data set that we fed them with. Like, they've got all the biases of the internet, and they've tried to smooth the rough edges off them, but you still see them. And like any tool, there's the whole dual use thing, right? It can be used for good, it can be used for ill. We've already seen, we've just had some elections over here in the UK, and there were deep fakes of uh, one particular politician, like, showing that he did something which he completely didn't, but it went
Oh,
wild
wow.
on social media, right? So I don't think when you're talking about powerful technologies like these, it's at all reasonable just to think about the positives of
Mm-hmm.
them. Um, but yeah, I just want to move fast. I want these things, these things these things that have the potential to do so much good. Let's do them already. You know.
Right. Yeah, and I mean, I'll just take a quick aside to this answer as well. I mean, it almost seems to me like there needs to be, similar to any new technology or new sort of experimentation any large pharma company, needs to be doing. It seems to me like there needs to be like an office of experimentation with all of these tools. What are the good things, that might help the company, and what are the things that might not? And I've seen, inside of large pharma companies, they, oftentimes will, you know, once a technology has reached a certain level of maturity, then they'll sort of, take it on. Um, but it has to get to the level of maturity where the chief scientist, chief technology officer, chief, CIO, or the maybe even the CEO of the pharma company comes back and says, well, why can't we do this better? And instead of waiting for that, I mean, why not have, I don't know. I feel like in the space that you're in, I feel like there are a lot of companies working on this same exact thing, and having a level of just somebody looking at all the tools that are available. It seems to me like the right approach to sort of see, hey, look, can we, I mean, to all of your earlier points, can we improve our output? Could we improve the actual impact that we're going to have on the patient? Could we improve, our time to getting things to market, all by, having, one of these? And the reality is the cost of them probably is far less than
Yeah.
than actually doing the real science in the lab. So
Oh,
yeah,
absolutely. But like, and companies do, right? They recognize this, and they try to put in a place, efforts to actually, try and make these transformations happen. But at the same time, like, completely understandably, they've had a lot of people come along making these kind of claims before, right? So, to your discipline of marketing, right, which is one that actually I've learned to have a huge amount of time and respect for. I think it's a fascinating art and science. And like, I, it's difficult to be we don't have the problem of not having something and having to hype it up. We have the problem of having something but having to cut through all the noise,
right.
um, and actually, connect with people in a way that they can see that we're both sincere and excited, and that yes, this could apply to their work as well, right? And that's a real challenge. It's a fun challenge, but it's a real challenge.
Yeah, absolutely. Last question: what excites you?
Yeah, a lot of things. I'm quite an enthusiastic person. Um, uh, I think that I'm genuinely quite optimistic, on the whole, which is a bit of a crazy thing to say when the world is in the state that it is at the moment. But I do tend to be pretty optimistic, and I think the 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 potential of humanity excites me. You know, in all seriousness, like I get to I'm pretty privileged in that I get to work with and do stuff with a particularly cool slice of humanity. I think, but like I think that it's very easy to get bogged down in like the day to day of the news cycle, and this election or that election, or this conflict, or or these economic problems or whatever. But overall, I believe very much in the capacity of people to innovate and to make the world better. And I really hope that I turn out to be correct, um, and that I'm not actually optimistic. I'm turn out to be realistic.
Well, Marcus, thank you so much for sharing that. And thanks for being on the Life Science Success Podcast. I really appreciate you being here and for telling us about Synthace and everything that you're working on. I wish you all the luck in the future. And yeah, it'd be great to connect as you continue to evolve.
Don, likewise, thanks for having me on.
Thanks so much.